welcome back to the 23rd lecture. So, today we are initiating lecture 23 and this will be monoclonal antibody production using phage display. So, in the last class we talked about monoclonal antibody production via hybridoma technique. Just to recap about the step before we move to the next technique. So, the first step in hybridoma is the process of fusion of B cells expressing unique antibodies with myeloma cells, creating a hybrid cell line called hybridoma. Clone screening, identification of the clonally derived hybridoma cell lines which are producing high amount of monoclonal antibodies. Because remember one thing, why this clone selection is important, it is not necessarily all the splint cells or B cells which are produced will have the equal propensity of producing monoclonal antibodies. Some of them will produce more, some of them will produce less. So, for higher productivity, we will have to pick up the ones which produces more monoclonal antibodies, and that is why this particular step 2, what you see, clone screening and selection is of extreme importance. Part 3 is cell growth. Cell growth is determined by monitoring the cell division over a given period of time using level pre-imaging. Step 4, specificity and cross-reactivity. This is basically the verification route, the process of analyzing the ability of an antibody to bind only one target antigen in order to prove its monoclonal ability. Binding affinity internalization. Binding affinity is the strength of the binding interaction between single biomolecule, that is protein or DNA, to its ligand binding partner, drug or inhibitor. And internalization is the process of monitoring the ability of a given particle to enter inside the cell. So, these screening processes are extremely important for monoclonal antibody production via hybridoma technique. So, today's class, which is class 23, will be on monoclonal antibody production via phage display. So, as I mentioned in the previous class, this technology is slightly more molecular biology intensive. In this method, the variable heavy chain and light genes of an antibody are introduced into a bacteriophage coat protein gene, resulting in phages that display the antibody on their surface. So, let me repeat it. In this method, the variable heavy and light genes of an antibody. So, if you look at the structure of an antibody, it has heavy chain and light chains, and there are genes which exclusively coat or code for the heavy chain and the light chains. Okay. So, what you are doing here is, now let me again repeat, the variable heavy and light genes, or the genes which are involved in the expression of variable heavy and light chains of an antibody, are introduced into a bacteriophage coat protein gene. So, you are basically introducing those genes, you are cutting that part of the DNA and introducing it into the bacteriophage protein gene, resulting in a phage that display the antibody on the surface using this strategy. So, automatically this bacteriophage is going to express it. So, you will be seeing the signature of the antibody on the surface of the bacteriophage. So, it is kind of a phage display technology. Using this strategy, scientists can generate an antibody library displaying millions of different antibodies. These displaying phages are then screened against the antigen of interest, and those that are highly specific are identified, isolated, and further characterized for downstream work. So, what you are essentially doing, for every antibody, there are genes which are coding for those antibodies. It has heavy chain, it has light chain, it has a variable heavy chain, variable light chain, so on and so forth. So, what you are trying to do is that you pick up that fragment of the DNA and you are integrating that particular DNA into a external code protein gene of the bacteriophage, and you are expressing them. So, what you are seeing on the top of the bacteriophage is expression of those antibodies whose genes you have integrated into the bacteriophage system. So, you have a display on the surface of the bacterial cell or bacteriophage, whereby you have a better window to screen for wide range of antigens. Okay. So, now how it works is something like this. You have a panning process using plate washer, then you have colony picking, step 2, then you have antibody interaction or affinity interaction, and then the functional screening. And of course, you have all the different technologies in order to rapidly do the process. So, panning is an iterative process for enriching phage within a population that possesses high affinity binding to a target of interest compared to others. Begin by enriching your population of phage with high affinity binding by exposing the library to your antigen of choice and then eluting and amplifying only those with highest binding affinity. If you remember, while we are talking about the hybridoma technology, I told you that while screening, you look for those particular hybridoma cells which produces highest proportion or highest concentration of antibodies. So, here also something of that sort you have to do, which has the highest binding affinity. Then comes the step 2, which is colony picking. Bacteriophages selected from the previous steps are then cloned and picked in order to isolate each unique protein binder. This is this step, colony picking step. Okay. So, we talked about the panning step, now we are into the colony picking step. Now comes the third step, where antigen antibody interaction, AG antibody, this is AG stands for antigen antibody interaction. During panning, phages displaying protein with higher binding affinity are selected in relation to phages displaying lower affinity protein. So, this is that binding affinity assay. This qualitative selection process requires validation using more quantitative immunoassays to assess antigen antibody interactions such as ELISA, immunofluorescence, HTRF, complement fixation, agglutination, and precipitation. So, when you talk about binding affinity, so we are talking about with what force they are binding with each other. So, you need to have very specific assays or quantitative measure. It's something like one finger. So, for example, versus like this is much more stronger binding. So, unless you have a quantitative assay to figure out is it just 2 fingers are binding with each other, or 2 fingers are binding with 1, or 2 fingers are binding with 2, or 4 fingers are binding with 5, or the whole 5 fingers are binding with 5, you have to have a quantification. Without that quantification, these kind of selection processes cannot be validated in a long run. So, those techniques, quantitative immunoassays to assess our antibody antigen interactions such as ELISA, immunofluorescence, HTRE, complement fixation, agglutination, and precipitation. These technologies are the ones which are of prime importance in terms of quantification. Next comes the step 4, which is functional screening. Following the characterization of antibody antigen interaction, which is basically the binding interaction or binding affinity, candidate molecules are then screened for functional activity, that is viral neutralization, vaccine efficacy, often using cell-based assays. So, final game is, does it able to neutralize your antigen? does it able to neutralize the virus against which this antibody has been produced. So unless you do a cellular assay of viral neutralization, your technology will not be fully vetted. So the vetting can only happen when you have a neutralization assay saying that, okay, this much amount of antibody neutralizes this much amount of virus. Why this is important from biosensing perspective? Because when we are talking about a reaction on a chip, we should have a quantification. We should know how much amount of antibody or antigen you are depositing on the top of the chip plate. Because if you know the number of molecules, then you know how many opposite kind of molecules will bind to them and how much signature. If you remember, I told you that you can detect them using fluorescence assay, you can use them electrochemical assay, you can use SPR assay, you can use AFM assay. There are a series of assays you can use, but these are all signals. These interactions have to be quantified. So if you have an assay to quantify them, then you will be able to say, okay, for X molecule, I need opposite kind of Y molecules. And based on that, I will get this much signature or a signal. That's why these individual assays, these individual step, understanding the basic kinetics of the individual step, understanding how much fluorescence signals will come, how much SPR angle will shift, how much AFM will alter, how much quartz crystal microbalance changes we will be able to evaluate, how much electrochemical impedance is going to show up are of immense significance. So you understand, it's not only you need to understand the biology of the production of these antibodies, antigens, but you have to understand how you can detect them, how you can quantify them. So that's why the modern biosensing, even as a course, it is very challenging because you have to integrate everything together for your understanding that how critical each one of these steps are. And there is no one way you can quantify them. You have to use multiple ways to validate your result. And that's why I'm repeatedly telling we are heading into the era of multimodal detection. And this is the key. You have to understand that multimodal detection is changing the face of our understanding about several technologies. Okay. Now, if I summarize this whole thing, what I have talked to you, we talked about hybridoma technology, we talked about display technology, then of course, included with is single B cell sequencing and next generation of protein sequencing. So here's the animal, target antigen immunization. In hybridoma, repeating it is splenocyte harvest, fusion with myeloma cells, hybridoma generation, and molecular antibody candidate. This is the cellular technique, hybridoma. Then you have the display technology, where you have B cell collections, mRNA extraction, RT-qPCR, isotypes of specific PCRs, phage or yeast library generation, and molecular antibody or monoclonal antibody candidates. While we are doing monoclonal antibody candidate, you have single B cell sequencing, PBMC's collection, FACS, fluorescent assorted cell screening, next generation sequencing assays, you have the MAB candidates. Similarly, you have next generation of protein sequencing, where sera or plasma collected, supernatant and affinity purified sample, sample processing, then you go through LCMS, MS, then you do a sequencing, and you have the MAB candidate. So you look at it, there are multiple ways how you can generate monoclonal antibodies. And this is just a snapshot of all the technologies which are available at your disposal. And you have to have a fair understanding at least of these two technologies, because these two technologies, hybridoma and the display technology. These are kind of pillars in order to understand how monoclonal antibodies could be produced and could be utilized. Once you understand these two, then rest will follow. So, you are observing that much of these technologies are extremely interdisciplinary, and you cannot really uh, shy away from it, because you have to understand the production. Once you understand the production, you will have a better hang on the system, that how these systems are working. Adding up to that, I wanted to add this slide because there are a lot of terminologies which are coming up. So when we talk about, I was talking to you about antibodies, this is how the antibody looks like. It has the heavy chain, light chain, variable heavy chain, variable light chain. Now, there are terms like nanobody. If you follow this whole picture, you will realize then you have SCFV, there are bispecific SCFV, then you have nanobody SCFV. These are newer and newer terminologies which are coming, and whenever you read a paper, you come across and you get scared. Oh, I have no idea about this. But that's why I thought of putting this slide in front of you. This slide will kind of give you an idea what are chimeric and humanized antibodies, human antibodies, chimeric antibodies, like you are integrating mice and a human, you are making chimeras. Then you have tribodies, disbodies, mini bodies, camelid heavy chain antibodies, and then you have FAB fragment, scFAB fragment, bispecific FABs. Then you have immunocytokine, that is nanobody, FAB cytokine, diabody cytokine, and you have the interactions. Some of these terminologies, so that you do not get kind of scared. These are all newer terminologies which are basically from the fundamentals of immunology, that there are different fragments. You can isolate the DNA for a specific fragment, you can clone that, you can express it in using a Phage display or any other technique, as a matter of fact, and the molecules which are generated, they have a different kind of naming you are doing. So, do not get bogged down by the nomenclature of all these things. They are simple nomenclature, they are all over the literature, but what I will highlight for you guys to understand these two basic technology, hybridoma and the display technology or Phage display technology, because these are the two techniques which are of prime importance for you people. And of course, you should know some of these, or you should not get scared of these different names which are popping up, which will pop up time to time, that what is a nanobody, what is this, what I mean, it is, literature is kind of completely flooded with these kind of terms. Okay, so do not get scared about these different terms which are being used. So to summarize upon, before I move on to the next part of it, is to the basic slides. So this is where the hybridoma technology started, and even before. So this is where we started this class: immunization process, fusion process, or the preparation of myeloma cells, clone screening and picking, functional characterization, scale up and winning, and expansion. And somewhere down the line, we talked about this technology, where we talk about the panning, and this is basically your Phage display technology, colony picking, antibody interaction and affinity specificity, and functional screening. Okay. So, these are the basic 4 steps involved in it, and this is the summary sheet in front of you, where you have hybridoma, display technology, single B cell sequencing, and next generation of protein sequencing which are being used. So, as long as you know the framework of this whole process, you are good to go. And as I mentioned, there are multiple such words which will come. They should not bother you, they are fine, you can deal with them. So, You all can deal with these different words which are popping out, as long as you understand they are nothing but different variants of antibodies, different variants of molecular techniques which are used to express different part of the antibody, say varying low chain, variable low chain, variable heavy chain, or some other part of the antibody, and they are being given newer and newer name, nanobodies, these bodies, and likewise. So, do not get fumbled by these terminologies, but overall you should understand this is the scheme of things where you have hybridoma technology, display technology, and these are the potent tools for antibody production. And as long as you kind of keep these in mind, you are good to go. Okay. Thank you, and we will move on to the next class after this.